I'm Dr. Jose Kraveshil. In my last class, I discussed the tools of genetic engineering, namely, number one, DNA modifying enzymes, which include restriction endonuclease that cuts DNA, DNA ligase that joins DNA, DNA polymerase that replicates DNA. Second tool is vectors. In the last class, we discussed two types of vectors, cloning vectors and expression vectors. Today, I am going to talk about the characteristics of a vector. See, let us take the example of PBR322. In the last class, I told you it is a cloning vector, which means it was modified from an E. coli plasmid so that if we insert a gene inside it and put it into E. coli, we can make multiple copies of that gene. That is called cloning the gene. Okay, so it's a cloning vector. First characteristic of PBR322 is the region called ORI. Full form is origin of replication. Okay, now what happens in ORI is when you introduce PBR322 into E. coli, the bacteria E. coli, the DNA polymerase of E. coli binds to ORI and replicates the plasmid. Let us say, imagine this is E. coli. This is PBR322. That's a plasmid. DNA polymerase of E. coli binds to ORI and makes multiple copies of PBR322. Okay, so that is why it is called origin of replication. Now, adjacent to the origin of replication, there is a region called rope. Full form is repressor of trimer. So, the function of rope is to produce proteins involved in the replication of the plasmid. So both ORI and rope are involved in replication of the plasmid to produce multiple copies of the plasmid in E. coli or bacteria. Okay, now the second character is selectable markers for transformation. Selectable markers for transformation. So here what we are trying to do is we get three types of E. coli when we do the experiment involving gene cloning in bacteria. We get those E. coli which have the nucleoid in it, but it does not have PBR322 inside it. That's one. In the second one, this is nucleoid, you have PBR322, but it does not have the gene inside, foreign gene inside it. Okay? Third one. You have nucleoid, you have PBR322, and you have the foreign gene. Now, to distinguish between these three bacteria, this is called non-transformant. These two are transformants. They are transformants because Both of them have a foreign DNA inside them. And to distinguish between the two, this is called non-recombinant and this is called recombinant. So we get three types of E. coli, one in which there is no PBR322 which is non-transformant, one in which there is PBR322 but there is no foreign gene, that is called non-recombinant, one in which both PBR322 and the foreign gene is there, that is called recombinant. We are interested only in the recombinant bacteria. In order to select the recombinant bacteria, we use two marker genes. One is the ampicillin resistance gene, the other is the tetracycline resistance gene. Okay. Now, short form AMPAR stands for ampicillin resistance gene. TET-R stands for tetracycline resistance gene. Now, both ampicillin and tetracycline are antibiotics. So, these are antibiotic resistance genes. So, PBR322 created by two scientists, Bolivar and Rodriguez, have introduced into the plasmid two genes from other E. coli. One is ampicillin resistance gene, the other is the tetracycline resistance gene. So what this gene does is, ampicillin resistance gene produces an enzyme that breaks down the antibiotic ampicillin. Tetracycline resistance gene produces an enzyme that breaks down the antibiotic tetracycline. So these are used as marker genes, which means they are used to select bacteria which contain the foreign gene, or we can say recombinant bacteria. That is why they are called selectable markers. All vectors should have selectable markers. So that's the second character. Now, the third character a vector should have are what are called cloning sites or restriction sites. These cloning sites or restriction sites are places where different restriction endonucleases cut the plasmid. Well, let us say we want to cut the plasmid inside tetracycline resistance gene and put the foreign gene inside it. So we can either use BAMH1, which is a restriction endonuclease obtained from Bacillus amyloidoliquefaciens, or we can use SAL1 obtained from the bacterium Streptomyces albus. So actually we need only one enzyme to cut the plasmid. But Bolivar and Rodriguez, who created PBR322, is giving us choices. We can use any of these enzymes to cut the plasmid. So our purpose is to cut the plasmid, insert the foreign gene inside it. Let us say we are going to use BAMH1. So what will happen is, if this is PBR322, this is the tetracycline resistance gene. We are going to cut here using BAMH1. So BAMH1 is cut here, will cut here, and we introduce the foreign gene. That the gene, this is the gene that we are going to multiply inside E. coli. We introduce the foreign gene inside it. So what happens is, This is that R. This is the foreign gene which is introduced by cutting with BAM-H1 here. So once foreign gene enters the plasmid, that R gene becomes inactive. That is called insertional inactivation. I'll explain once more. Insertional inactivation is the inactivation of a gene when another gene is inserted inside it. We are going to learn the details later. Why we use insertional inactivation. Okay. Now, cloning sites or restriction sites are places where different restriction endonucleases cuts PBR322. Let us look at the sources of the different enzymes. Now, PST1. PST1 is obtained from the bacterium Providentia stewarti. Providentia stewarti. PVU1 is obtained from Proteus vulgaris. Eco R1 is obtained from E. coli. CLAV1 is obtained from Caryophyllon latum. HIND3 is obtained from Haemophilus influenzae. Now, BAM-H1 is obtained from Bacillus amyloliquefaciens. SAL1 is obtained from Streptomyces albus. PVU1, we discussed PVU1 here. This is PVU2, is obtained from Proteus vulgaris. So these are the sources of the different restriction endonucleases that cut the plasmid. We need only one enzyme, I told you, because we are going to cut the plasmid at one location and insert the gene inside it. One thing you have to remember is, one enzyme should have only one site. I'll tell you the reason why. Let us say, this is PBR322. Let us imagine a situation in which there is a site for BAM-H1 here, another site here, and another site here. Let us imagine there are 3 sites in it. What will happen? This will be cut into many pieces. So, in order to avoid that, one enzyme should have only one site. So, I'll repeat once more. A vector should have an origin of replication, selectable markers for transformation, which are used to select bacteria which contain the foreign gene. And number 3, several cloning sites or restriction sites for several restriction endonucleases, but one enzyme should have only one site. These are the 3 characteristics of a vector.